Now we begin with the session 2. And I would like to invite our chairpersons for the session link between the gut microbiota, probiotics, and inflammation. Professor V. Samuel Raj, Director and Dean Academics, SRM University, Delhi, NCR, Sonipat, India, and Professor Debajyoti Banerjee, Professor and Head, Department of Experimental Medicine and Biotechnology, Postgraduate Institute of Medical Education and Research, Chandigarh, India, to hand over the session. Can we have huge round of applause for Professor V. Samuel Raj and Professor Debajyoti Banerjee? I request Professor V. Samuel Raj and Professor Debajyoti Banerjee to start the session.
Thank you very much. Thank you very much. Now we will start the session 2. The first speaker for this session is Dr. Wong Hei Sunny. Dr. Sunny has done his PhD at the University of Oxford. Currently, he is working as a dean, assistant dean, and program director of microbiome medicine at the School of Lee Kong of Medicine, Singapore. Dr. Sunny is an expertise in gastrointestinal diseases and gut microbiome. His main research interest is on the host microbe interaction in digestive and systemic diseases. Dr. Sunny has won several awards, including this Asia Pacific Digestive Week, Emerging Leaders Lectureship, the Sir David Todd Lectureship, and the Croucher Foundation Award. He is currently an associate editor of the Journal of Gastroenterology and Hepatology. Now I invite Dr. Sunny to deliver the talk on exploring gut microbiome, a link between obesity and cancer vulnerability.
Thank you, Professor Raj. And may I confirm that the screen and my voice are all right?
Yes.
Yeah, thank you very much. And first of all, I would like to thank Professor Raj and also Professor Benabi for the introduction, and also the organizer for inviting me to give this talk. And it is a great honor. And also, I apologize for not being able to be there due to my visa issue. I very much want to be there because actually last year at around this time, I was in Chandigarh, and we had a great time. You can see me that we visited the hospital and also different places. So I really enjoy it. So I was looking forward to come, but unfortunately, due to some visa issue, I couldn't make it. But nevertheless, I'm really happy to see you here. And my talk will be on microbiome and as a link between obesity and cancers. I guess when we look at this topic, we are looking at three things over here. So we know that we have a lot of metabolic conditions. So those would include common things like obesity, diabetes, and fatty liver, or sort of other cardiometabolic conditions. We know that they are one major risk factor for cancer. So, and it increases the risk of many cancer. So we want to know how does metabolic conditions affect these cancer. And now microbiome comes into place. So we know that, for example, microbiome does contribute to a lot of different cancers as well. And I think it certainly play a role in metabolic condition. So I think the essence of my talk would be on the triangular relationship between these three. And importantly, how does microbiome play a part in the increasing or affecting the propensity where metabolic conditions predispose to people with cancer. So first, when we look at metabolic conditions and cancer, so this is a chart that actually can see the relationship. And obviously, I think it is very clear that metabolic conditions increases the cancer risk. You can see that they are slightly different. So there are different conditions. Here we are looking at obesity, and there are different cancers as well. But all across many organs, you can see that the risk is increased. And so here, I think it was listed at least 13 organs. I know that it could be more, although these 13 are the ones that are more accepted and also proven. But that doesn't mean, of course, that the risk on other organs are not there. But when you look at this chart, you can see that a lot of them are gastrointestinal cancer. I am a gastroenterologist, so I'm very concerned about this cancer. You can see that the gastric cancer is there, the colon cancer, esophageal cancer, and pancreas, which is increasing. So they are almost all increased in people with obesity. You see that the top one on the chart is esophageal cancer. We know that there are a few types. There are squamous and there are adenocarcinoma, and particularly adenocarcinoma, which occurs more commonly at the lower esophagus, is related to the acid reflux that causes inflammation, injury to the lower part of the esophagus, and therefore the risk is very high in this. And I think the other part that is quite high is the uterus. And it's due to the hormonal effect from the cancer. But all across this cancer, you can see that the risk has been increased quite high. So when we look at a bit more recent study, there were also this study looking at over 300,000 people with the UK Biobank. Over 100,000 of them has metabolic syndrome, and they look at the incidence rate. But comparing to people who do not have this metabolic dysfunction, you can see that the risk of cancer is increased. And you can see that most of the odds ratio are between 1.2 something. So meaning that the risk is roughly about 30% more compared to people without the cancer. So the risk is quite definite from all these studies. And I think the other factor is to look at the proportion or the characteristics of this risk. So given that people with obesity or increased BMI, they have a higher cancer risk. Also, the next question that you would ask is, how is the risk? Is it linear or is it exponential? And there are also a few studies looking at that quite recently. And you can see that on the right, you can see that the relationship is actually a U-shaped relationship. So there are two stories that, or two messages we can get. One is that there seems to be an optimum that at around the BMI of about 20 something, that the risk is lower. So between 20 to 24, the risk seems to be optimal. Where if you are a lot slimmer, actually the risk increases. It doesn't decrease. So this is a U-shaped relationship. The other thing that you can see is the right tail, that it is a linear or almost more than linear relationship. So if you increase your BMI, the risk of cancer, relative risk increases. But one important message is that it doesn't plateau or stop, at least till the end of the graph at about 40 to 45. So the risk seems to be still increasing. So it doesn't actually stop. So one message is that when you are obese, the more obese you are, actually the higher the risk. So this is the current understanding. And then there are also studies looking at what we call metabolically healthy versus unhealthy obesity. So meaning that people have increased BMI, but that they have less metabolic dysfunction when they look at their glucose, their lipids. So you can see that on the chart there that they still actually at a high risk as well, although they're better than people who are metabolically unhealthy and obese. But I think the increased risk is still observed in some of these epidemiological studies. And these are the pattern of characteristics that we see. A bit of mechanism. So the obvious one question is, why does obesity increases the risk of cancer? And this is not new. So there are classical mechanisms that attempt to answer this question. You can see that insulin signaling is important. So we know that insulin and the IGF-1, for example, they are mitogens, so they can increase cell proliferation. So this is one important mechanism. Second one pertains to the adipose tissue hormones, namely the leptin and adiponectin that are secreted from our adipose tissue. And we know that they can increase the inflammation and also promote inflammation and also cell proliferation changes. And then the sex hormone metabolism, namely the estrogen and also the estrogen receptor, could also mitigate some of these cell changes and also increase risk of cancer. But apart from this classical mechanism that we understand for that case, there are also newer ones that we look at. Information is important, so this is the theme of this section. But basically what we know is that people with metabolic dysfunction, diabetes, obesity, and all alike, they are characterized by chronic systemic low-grade inflammation. And we also understand that this inflammation seems to derive from gut microbiome-derived endotoxin. They are the LPS that seems to go from the gut through a leaky gut into the blood, and that perpetuate the inflammation there with increased level of CRP, interleukin 6, TNF alpha, all around our body. And this inflammation in over the long term could increase the cancer propensity as well. There are also other newer mechanisms. You can see here that gut microbiome is one that we will talk about, obviously, but also GI hormones, also modulation of extracellular matrix. So these are other mechanisms that has been proposed to mediate the effect of obesity-related cancer. You can see that, for example, some of the hormones, including neurotensin or even CCK, they has been shown to be proliferative as well in cellular studies. So about the microbiome, so we know that actually cancer is of course caused by genetic and environmental change, but we know for some time that actually microbes play a big role. And these are what we understand. Helicobacter pylori, obviously for gastric cancer, a lot of viruses that could promote liver cancer and other cancer as well. So these are known cancer pathogens that we know. But the concept of microbiome also come in with an ecological concept. So that is what we see. For example, in colon cancer that I also work on, so we see newer bacteria that has come up. So bacteria such as Fusobacterium, Bacteroides vajalis, they has been shown to cause cancer. And we did some of these studies to show that they appears to work in a network of ecology. And this network appears to be actually promoting cancer formation in the colon. Gastric cancer is another example. So apart from Helicobacter pylori that you see is a major carcinogen, there appears to be other bacteria that could increase the risk and also act synergistically as well. We also know in my previous lab, there was a recent paper looking at Streptococcus aeruginosus that can promote and causes gastric cancer. And it might be the second bacteria after Helicobacter pylori that has been functionally shown to increase carcinogenesis in the stomach as well. And we mentioned about cancer microbiome, and we know that obesity in people with metabolic disease, they have a change of microbiome. So on the left, you can see that it's a classical studies where people were obese. When they took the microbiome and transplant into animal, that the animal would copy the phenotype of that donor microbiome. So you can see that the mice, for example, increase in the weight, and with a right diet, high fat diet, they kind of increase their weight and also increase in the adiposity, showing for the first time that the microbiome itself is transmissible, and also obesity in a way could be environmental and can be partially transmissible by transplanting the microbiome. So it was an older study, but what from the right you can see are actually a lot more studies looking at the association between microbiome and obesity. One thing that you can observe is that actually among all these studies, because they are from different places, different continents, and different study design, they all have very different microbiome, but almost across all of them, they see a difference between people who are obese and people who are normal in the weight as well. And there were also studies that look at trying to attempt to look at a consensus microbiome signal across these studies and continents, and also to look for obese microbiome signature. So looking at these mechanisms, so these are the microbiome related mechanisms that we have talked about, and there are quite a bit of being studied. So you can see that this altered change in microbiome is associated with increased inflammation, increased endotoxemia, LPS level, and also gut permeability changes. And on the right, you can see that they are also associated with changes of certain metabolites, such as short-chain fatty acid and also bile acid, that can itself promote cancer formation. And we will talk on some of these later. So we mentioned about metabolic associated cancer. So there were two examples in liver cancer that I would quote here. We know obesity alters microbiota, and this microbiota has been shown, for example, to increase the level of deoxycholic acid, which is primary bile acid. So we know bile are produced by the liver, but what is also as important is that the gut microbiome in the colon, in the gut intestine, could actually metabolize and also change some of them. And some of them can actually be carcinogenic. So for example, in here, this example of DCA, and it has been shown that it can be absorbed and recirculate through the enterohepatic circulation and go to the liver to stimulate the stellate cell to secrete inflammation factor that promote inflammation, fibrosis, and proliferation of the cell, and therefore increasing the risk of the liver cancer. This is another study. So on the right, you can see that what we mentioned, modifying the bile acid that were modified by the gut microbes, they could change the portion between primary and secondary bile acid that changes the cancer propensity. And on the right, there's another mechanism that has been proposed where the microbiome could suppresses the NKT cell that usually carry out the immunosurveillance against the cancer, and through the CXCL6 mechanism, then eventually reducing these immunosurveillance and increasing cancer propensity in the liver as well. So these were two major mechanisms that has been proposed of how gut microbiome and also the metabolites could be related to formation of liver cancer. We also did some study, and our primary interest is on colon cancer. So our research question in this study is whether obesity-associated microbiome could promote the occurrence of metabolic or obesity-associated colorectal cancer. So actually, we took a transplant model. So what we did was we took the stool that has the microbiome from obese people, and we also took some from people who have a normal BMI, and we put them into cancer model in animal. So we did 3 cancer model. We did what you can see here, AOM, which is a carcinogen that we add into this animal. But apart from this, we also did an APC genetic model, and also we did an autobiotic germ-free model as well. But what you can see is all across these 3 models, in people, in the mice that receive the obese microbiome, so the microbiome from obese people, they all uniformly has more tumor formation. So what we observe is that they have more tumor, they have a higher tumor load, and also there are more dysplasia and also cellular changes, including Ki67 proliferation marker in these animals. We also saw changes in the gut barrier dysfunction and also increased inflammation. And we look at them under the TEM microscope. We see changes with the tight junction as well. We saw changes in oncogenic pathways, especially with the Wnt signaling pathway, and also we were able to reproduce some of these findings looking at the Western blot at the protein level as well. And importantly, when we look at the microbiome, that we see that the microbiome change persists in these animal beyond 24 weeks as well. So they do give a different microbiome signature, and there were also microbes that show changes that correlate with the tumor formation as well. On the right are some subsequent in vitro work where we also look at a xenograft model, and also we took it to in vivo model and show that some of these bacteria, for example, AF is Alistapi fingodi, that could be associated increased tumor formation, whereas some bacteria such as Akkermansia, that could be associated with a suppression of the tumor weight. So I think this is very interesting. And apart from our study, there were also studies showing relationship of this obesity microbiome and also with cancer formation as well. So for example, in this study that was recently published, what they did is, again, they also gavage stool from obese and versus lean people. But what they observe is in the adipose tissue, they observe increased changes in some of the neutrophil subtype. And these neutrophil subtype express transcriptome that shows a pattern that is compatible with colon cancer, suggesting that this obesity microbiome can induce inflammation, changes in neutrophils, and also a signature that corresponds to a cancer phenotype. So lastly, I would like to take a few moments to talk about diet, because diet is extremely important. So we know diet, of course, is important to our health, increasing, and that includes cancer risk as well. We know, for example, colon cancer is highly related to consumption of red meat and also processed meat. So in a way, this is extremely important. And now we know also that diet is one of the most important factor that affects the microbiome. So the role of microbiome is also definite, looking at this diet health relationship. And basically, we are what we eat. And the moment that we are eating, our microbiome are eating as well. So this is how important they are. And to convince you, so you can see that, for example, what we eat in a very rural context is very different from what we eat in a modern city. You can see that the chicken on the left is running behind. There's a lot of greens, and also all the food that this person eats is actually directly fresh from the bag. Whereas on the right, you can see that they are highly processed. They are, in a way, cooked, and also you can vaguely see the M sign here that signify a prototypical example of fast food. And this is a picture of what I eat in Chandigarh last year. And surely with this different diet, the microbiome would be very different as well. So there were studies looking at this relationship between diet, microbiome, and cancer risk. So for example, in these studies, actually two studies are almost about 10 years apart, but looking at a dietary swap among African Americans and rural Africans. So they were genetically similar individuals, but if they swap the diet, what they observe is even despite a 2 weeks swap between kind of rural versus a very westernized diet, they see obvious change in the colonic marker of cellular proliferation. Of course, 2 weeks of diet, they don't see a cancer, but they were already able to pick up cell proliferation marker that correlate or surrogate as a cancer risk. And on the right, that you can see profound changes with the microbiome and also the metabolite, including some of the bile salt that we mentioned. We know that, for example, high-fat diet also changes the microbiome. So in these two studies, you can see that they change the microbiome profoundly, involving changes with Parabacteroides that are associated with fiber, and also Alistapes that are associated with high-fat diet. And on the right, you can see that these changes could lead to different metabolites such as inosine that were described there, and also increases the cancer risk in vitro and also in vivo model as well. We also did some studies. We know that there are studies, for example, in the UK that look at the PREDICT cohort. What they did is they look at 1,000 people looking at the diet and microbiome. So there are obvious changes. And also what they observe is that some of the changes in diet and also the microbiome correlate with the inflammatory score in the body as well. So taking the opportunity of the Helios cohort also in Singapore, so this is like a national cohort. We also look at the microbiome and also the dietary history of these people. In here that you can see, for example, we can see obvious association between the food that they consumed and also the microbiome they have. And also we were able to correlate with the microbiome change and also metabolic conditions in these people as well. So I think finishing off, this is my second last slide. You can see that what we are looking is that obesity associated or metabolic associated cancer. There are different preventive strategies that are important because these are what we clinicians are concerned. You can see that, of course, we have strategies such as a diet, exercise, and all of these are important. But that also means that with the evolving understanding of diet microbiome relationship, there are also new strategies. You can see that here bariatric surgery is obviously important to control the weight in some of the morbid obesity people. And in this study, you can see that actually apart from the metabolic benefit, bariatric surgery can actually reduce the risk of cancer compared to the non-surgical control by actually quite a significant odds ratio as well, 0.68. Other example, for example, GLP-1. So there were also epidemiology study looking at GLP-1, and that apart from the metabolic benefit, it also associated with a decreased cancer risk in this cohort. And lastly, for example, some of the microbiome finding that we found that microbiome modulation could be a potential strategy to reduce some of these metabolic conditions and also the associated cancer risk. We also want to mention, for example, that there are quite a bit of work involving some of ours to look at probiotic or what we call biotherapeutics or next generation probiotic to mitigate the cancer risk as well. A lot of them actually are classical probiotic we observe involving a lot of lactobacillus. So for example, there are also a plethora of studies looking at these different various lactobacillus and also how they could prevent the cancer. And in this case, Pentarium and also indole-free lactic acid as well. We also did some work to publish and also looking at the new probiotic we observe. And in this case, for example, Streptococcus thermophilus, and we also look at Roseveria on how they can also potentially reduce the risk of colon cancer. On the right is a study that you can see with Akkermansia. I mentioned this not in the cancer context, but we know this is an important novel next generation probiotic that has been associated with a metabolically healthy state. And there are also, so for example, this one is actually approved as a food that can be purchased over the counter, but there were also clinical studies showing that this Akkermansia could be a potential in reducing overweight and also improving metabolic conditions in people as well. So ladies and gentlemen, I think I will summarize that microbiome plays an important role in metabolic associated cancer, and there's immense potential of using it to modulate by diet or also probiotic or biotherapeutics to reduce the risk of metabolic associated cancer. So I would thank here very much everyone as an audience and also my team in NTU in Singapore, my previous work in Chinese U, also the funding and importantly organizer, and especially Dr. Nija that has actually helped me a lot during organization of this conference. So thank you very much, and I welcome any question that you may have.
Yeah. Thank you, Dr. Wong, for your excellent presentation and linking this obesity and cancer. As you said that, it's very clear that diet matters. What you eat, it matters. And you have given a very good link between the diet and the microbiome. And also you have linked very well how this diet, the high fat diet as well as the obesity and the cancer. In fact, you have given a very good, the link between these aspects and also explained very well the classical mechanisms and pathways. Not only the classical mechanisms and pathways and the new mechanisms. And what are the microbiome related to the cancer? And what are the microbiome related to the obesity? And these microbiome mechanisms. Thank you so much for your presentation. Now I open this forum for the audience. Yeah, please give the mic.
The question is, if...
Sir, please introduce yourself and then you ask the question.
Javed Agrawala, I'm from IIT Ropar, Punjab. And my question is, if it's known that a particular gut microbiota or a particular species or strain, if they predispose towards cancer, so is it not possible that we can kill these microbes and eliminate the chances of cancer?
Thank you for your question. I think a short answer is that yes, it is possible. And I think I think yes. So in a way, there are bacteria that has been shown to cause cancer, and like Helicobacter pylori, that we know eradicating it can reduce the risk of gastric cancer as well. So at a similar token, you can see that they can be applied to other conditions such as liver and colon cancer. And I think this is what we and also everyone in the world are trying to work on. The difficulty, I think, they are not without challenge. It is because one is in classical pathogens, the effect size is a lot bigger or is a lot more clear cut. So, for example, for a long time, Helicobacter pylori is one of the most known and also the top. And so that's why eradicating that would work. But in some of the newer microbiome study, it takes an ecological concept where each bacteria might play a smaller role, and also the other is they work in interactions. So it is a bit harder. The effect size might be smaller, but I think this is possible. And I think the other is, of course, we don't have a very good strategy to change or kill one bacteria as well. We know that antibiotics, we kill a lot more bacteria, so it may not be only that bacteria is killed, and also we may take a long time to recover. So these are some challenges that we are thinking about, but definitely I think this is possible.
Dr. Wong, I'm Dr. Ganguly. Uh, we have also worked on cancer and microbiome, and what we see that many of the strains which are the cancers which are not responding to drugs, many of them can break the cancer drugs because of the glucuronidase system which we have in our body. But what we find a similar finding as you, that they process the metabolic pathways like sphingosine pathway which leads to ceramide, which ultimately stops the apoptosis and other breakdown processes, or the AKT mTOR, the PI3K AKT mTOR pathway which you showed, as well as the insulin, IGFI, and the GLP-1 pathway. So this is a very strong association, and what we are recommending now, that those who are not responding to this pathways, they may be given fecal microbial transplant. And so this is a very good study, and I congratulate you for strengthening this hypothesis. But I'm very worried that if I have a fat person in my house, I may also become fat.
Yeah. Thank you very much. Yes.
Hello. Thank you, Dr. Sunny, for a very nice presentation. I'm Dr. J.B. Prajapati from Faculty of Dairy Science, Anand. In the later part of your presentation, you talked about erkapensia and the trial with live and dead, both kind of cells. So, what was the difference in the two treatments? Do you recommend dead also, or live is essential?
Yeah, thank you very much. It's a very good question. And we know in that study, actually they have live bacteria and they have pasteurized bacteria, and show that actually they have similar effect, or if anything, the pasteurized one might have a better effect. The difference are that if they are dead, it doesn't mean that they are not doing anything. It just means that it is the bacteria. It may be the antigen, it may be the secretions that's already there, but they may not live there, they may not colonize and proliferate to produce more. But still, they could have an effect because of their antigens, the proteins that are already there. And the one common understanding might be that the antigens do play a big role because the antigen give the LPS, they can stimulate the toll-like receptor 4, receptor 2 as well. So I think it's also the belief that they mediate some of the important effects. And I think we need a bit more understanding, but this has been what we observed in that study. And there are very few of those studies, so I hope to see more of that.
Thank you.
Yeah, Mike, please. Yeah.
I am Dr. Sashikaran. I was the former director of the National Institute of Nutrition here in India. See, you showed one study where in Africans, where by shifting to a high-fat diet, there's a the proliferation marker of the Ki67 had gone up. Does that mean that the healthy gut microbiome actually also has an anti-proliferative effect?
In that study, so what they did was they recruit two types of people, African American and African ethnic Africans in US, and then they swap their diet for 2 weeks. So it is a very drastic swap in the diet. So in a way, it is quite a total dietary change, but only for 2 weeks. But what they observe is that drastic change of diet, they change the microbiome and they change the metabolism. Of course, they can't see tumor in 2 weeks, but what they did was they did colonoscopy before and after. And then, of course, it will be normal. It will be the same. You won't have a polyp or cancer in 2 weeks. But what they did was they took the cell and look at the Ki67. That is a cell proliferation marker. Of course, it doesn't mean that they cause cancer, but in a way, it is a surrogate marker of how cell turnover, and in a way, it is a surrogate marker as well. But they did observe obvious change in terms of increased proliferation in the group that consumed Western diet. I think microbiome, they certainly have different effect on the cell proliferation. So in a way, short answer is yes. Some of them might affect the cell proliferation. Some of them might even suppress them. And of course, I think that is only a surrogate. That doesn't mean that they will develop a cancer, and you can't actually infer that it necessarily will represent a cancer in long term.
The last question for this speaker. Yeah, please.
Hello, Dr. Sunny. Thank you for your talk. I have a question regarding H. pylori. This H. pylori is present in a lot of people here in India, but it does not cause like gastric cancers in everyone. So does the mere presence of the bacteria itself can be associated with a particular class of cancer, or is it there something should be happening transcriptomically, or they should be producing some metabolites to cause cancer in particular populations? Or can we associate this gut microbiome dissociation with some genetic predisposition towards development of some cancers?
Yeah, very good question. Thank you. The short answer is yes, you're completely right. Because we know that H. pylori actually is the most important etiology of cancer. So it causes what we believe to be 80 to 90% of all the gastric cancer. But at the same time, we know that it doesn't necessarily cause cancer in everyone. So there are bacteria and also host factor. Bacteria factor being that some strains are more carcinogenic in a way, Cat A, Vec A, so some of the pathogenic or kind of the virulent strain associated, so they cause a small cancer, and also on the host. So for example, IL-1B gene is one that has been described to be associated as well. And then the host bacteria interaction, and some also talk about the mismatch, the genetic mismatch between the bacteria and the host that would increase the risk of cancer as well. And what is also interesting is that Helicobacter pylori causes a lot of cancer. It promotes what we call chorea carcinogenesis, so it increases intestinal metaplasia, atrophic gastritis. But what is interesting is that at a certain stage of intestinal metaplasia, even if we kill the bacteria, the cancer risk is still increased and is still there. And that's why people start looking into other microbes. And it has been shown that the other microbes also play a role. And that was the basis of how people look at other microbes as well as the Streptococcus aeruginosus that we describe and could be a player in the cancer as well. So all of what you mentioned are correct, and this is our current understanding.
Dr. Sunny, just one more question.
Hi, I'm Dr. Manu. So my question is that you touched upon the bariatric surgery and as well as the GLP-1, right? So in the current studies, is there what is the change in the microbiome? We all know that, it will reduce the obesity. That is the intervention which is being done pretty aggressively. But GLP-1 has caught the fancy of, a lot. But have there been studies and to what extent does it reduce the cancer risk in both the cases and the changes in the microbiome or microbiota?
Yeah, so this is very nascent and new. So a lot of GLP-1 studies, of course, are on metabolic. So people look at metabolic, and now people look at cardiovascular, renal outcome, and show that it is beneficial as well. And then people look at other things, including dementia, including brain conditions, and they appears to be a magic drug so far. And also, I think there are also new studies looking into the cancer risk. And what the paper I've shown is, I think, one of the very few. So I think there were two. There was another one that I looked at that described a low cancer risk, but there was an epidemiological study that looked at and compare people with GLP-1 and without, and they reported a low risk in colon, but also other cancer as well. So I think, but when you look at the mechanism, when you look at animal, not so much. I haven't found many. So I think there yet to be a lot more understanding. What we know is GLP-1, apart from the metabolic effect and also is a gut hormone, it affects the neurological system, especially on the hypothalamus, and it also affects inflammation in the body, including the gut as a part as well. So I think a lot yet to be understand in its role to cancer, but I think it has good potential.
What about bariatric surgery?
Bariatric surgery, we know that it affects the microbiome a lot. But of course, I feel also microbiome is not everything. So the other mechanism, including adipose tissue, including insulin pathway, they are changed as well. And it might well be that they also interact with microbiome. So it's also a collective of these biological mechanisms together.
Thank you very much. I know that there must be more questions, but considering the time that we have to move on, thank you, Dr. Wang, for your the excellent presentation, and also thank you so much for the audience for asking very good questions. Now over to my colleague, Dr. Banerjee.
Thank you. Thank you.